Welcome back. My name's Ralph DeBerardinis. I'm the Chief of Pediatric Genetics and Director of the Genetic and Metabolic Disease Program at UT Southwestern. And we're glad to have you here for the presentation of the 2024 recipient of the Mary Beth Maddox Award and Lectureship, which were established in honor of Mary Beth Maddox, former Executive Director of TAMEST, who passed away in 2018 after a valiant battle with pancreatic cancer. I had the honor of serving as the chair of the selection committee this year, and I wanted to thank my fellow members of the committee, which you may see a picture of them coming up here, possibly. And if not, you'll see it again because it's been on circulation today. So the award and lectureship recognizes women scientists in Texas bringing new ideas and innovations to the fight against cancer. And the recipient receives a $5,000 honorarium and the opportunity to present their work here at the TAMEST annual conference, and you'll see that in just a moment. In addition to being honored at the TAMEST annual conference, the recipient gives lectures across the state at TAMEST member institutions with NIH National Cancer Institute designated cancer centers. So this is really a distinctive and I think unique award. And I must say the committee this year had a very challenging task of selecting a winner from a very competitive field. So I want to thank all the nominators and the nominees for participating in the program this year. This year's recipient is Dr. Nidhi Sahni. Dr. Sahni is an associate professor in the Department of Epigenetics and Molecular Carcinogenesis at the University of Texas MD Anderson Cancer Center. And I'd like to now introduce the nominator of this outstanding scientist, Dr. Anil K. Sood. Dr. Sood is professor and vice chair for translational research and a leader in several other positions at UT MD Anderson Cancer Center. Dr. Sood is a TAMEST member from the National Academy of Medicine. And Dr. Sood, congratulations on your recipient.
Thanks.
I didn't know you were behind it.
Thanks. Thanks very much, Ralph. Dear colleagues and esteemed guests, it is with great pleasure and admiration that I introduce Dr. Sahni as the 2024 recipient of the Mary Beth Maddox Award. From her humble beginnings to her current standing as a true luminary in systems biology, her journey has been nothing short of inspirational. After finishing her undergraduate work in India, Dr. Sahni obtained her doctoral degree at the University of Iowa in biology. During this time, she worked mostly on infectious diseases and was particularly interested in developing approaches for preventing acquired infections. After that, she moved to Dana-Farber for her postdoctoral work, where she developed a keen interest in cancer systems biology and precision medicine. During this time, her mentor, Dr. Susan Lindquist, unfortunately developed ovarian cancer and subsequently passed away from it. In my conversations with Dr. Sahni, these tragic events had a transformational impact on her. Dr. Sahni joined MD Anderson in 2016. Inspired by her mentor's death, she has dedicated to systems biology approaches to develop combination therapies for ovarian cancer. While PARP inhibitors are widely used for treating patients with ovarian cancer, many patients eventually develop resistant disease. Dr. Sahni's research has focused on biomarkers that are key mediators of DNA repair, which has implications for screening, risk stratification, and developing new treatments. She pioneered the field of functional variomics, which is a field that aims to identify mechanisms by which DNA sequence variations affect diseases. Her work integrates experimental platforms, genome editing, proteomics, and next-generation sequencing to provide insights into cancer progression. Her lab has focused and has developed robust bioinformatics and computational algorithms to identify novel therapeutic targets. These insights can be used to develop personalized prognostic and treatment strategies for patients with multiple types of cancer. In addition to her accomplishments, Dr. Sahni is well-grounded in life. I asked her what she cherishes the most about her education and training. Her answer was that she really cherishes meeting her husband during graduate school and is highly devoted to her family. Please join me in congratulating Dr. Sahni on her remarkable accomplishments that paved the way for her selection as the recipient of the Mary Beth Maddox Award.
Okay, we'll now have a 20-minute talk from Dr. Sahni about her research, followed by a few questions.
Thanks. Good morning, everyone. It's really a pleasure to be here at this conference. I was attending, and I've met many people that I met at the past 2 years when the conference was held in San Antonio and in Houston, actually, where I work. And so it's really a great honor and privilege, and I'm really grateful to the committee for having selected me out of a really competitive pool of applicants, and really grateful for the opportunity to present my work here. So as Dr. Bernardinez and Dr. Sood mentioned, a large focus of my program at MD Anderson has been developing these approaches, what we call functional variomics approaches, as you'll appreciate as I go over the course of this talk, and how we can accelerate AI in biomedicine using some of these approaches. So just trying to see how we, oops, where's the advancement? Oh yeah, okay, got it. So just giving you a bit of a background of where I actually started, my initial training, and then, would really like to acknowledge, and I've been very fortunate to have met really amazing mentors along the way as part of my scientific career so far. So as Dr. Sood mentioned, I did my undergraduate and master's training at University of Delhi, which is my hometown, the capital of India. And thereafter, I moved to the US in 2005 to undergo my Ph.D. training at University of Iowa. And from thereafter, I moved from Iowa in 2010 to do my postdoctoral training between actually two labs, one at MIT and the other one at Harvard Medical School. And then I was very fortunate to have come to Texas in 2016 to start my faculty position at MD Anderson Cancer Center. So my early career, as Dr. Sood mentioned, has been in infectious diseases, and I was very privileged to have worked with a pioneer in Candida albicans biology, David Saul, at the University of Iowa. And that's where I started developing an interest in computational biology. And back in the day, in the early 2000s, as many of you in the audience will appreciate, microarrays were just starting to burgeon. And I was very fortunate to have done these microarray experiments as part of my PhD training and developed the expertise to do computational analysis, which then paved my way to really go into my postdoctoral training at Harvard. And I was very fortunate to work with David and was quite productive during my PhD career, and I published a number of seminal papers in Candida albicans infectious biology, and I was very fortunate thereafter to transition from my PhD at University of Iowa to go to my next training, which is where I was really honored and privileged. And I think I really want to dedicate this talk in memory of my amazing mentor, Susan Lindquist, who unfortunately passed away shortly after I moved to start my own lab at Harvard Medical School. And Sue was really an inspirational mentor and a strong advocate for women in science. And I'm till this day really privileged that I had a chance to cross paths with her when I was at the Whitehead Institute. And of course, an equal privilege goes to my other mentor, co-mentor, Dr. Mark Vidal, who's a pioneer in cancer systems biology at Harvard Medical School. And Mark and I still interact quite collaboratively on a number of initiatives and a number of consortia projects till this date. And after that training, which was really amazing, I moved from Boston and was recruited to MD Anderson Cancer Center by Dr. Gordon Mills, who's a giant in systems biology. And I was really fortunate that he was here with this vision and one of the earliest systems biology departments in the country. And I was recruited as a secret recruit to MD Anderson. And as I was talking to a number of people last night over dinner, I think CPRIT has been transformational in my career, and it really enabled me to take high-risk, high-reward type approaches in my research, and I've been forever grateful since coming to Texas to be able to take those kind of risk approaches. And now moving on to the science. So as Dr. Sood mentioned, a big portion of my lab works on understanding how genetic variation impacts human disease. And in order to be able to understand how genetic variation can contribute to disease, we first need to establish a framework for making these networks of interactions that happen in the reference cell or in the unaltered condition, which is a wild-type condition. And so we've developed this approach, what we call functional variomics, in which we can really assess the effect of all types of genetic variation, be it coding, non-coding, and even larger structural rearrangements, such as gene translocations that happen in cancer, and establish these libraries, huge numbers of these variants. We can really establish libraries of thousands of these variants and really assess in a high-throughput manner how do these variants impact disease biology in the context of cancer and, of course, many other rare genetic diseases. And so in order to study these, the impact of genetic variation on disease, we, of course, want to establish a reference interactome, if you will, of interactions that we can go after and then really profile what happens when we alter that gene. And so, when I started my work at Harvard, when I was a postdoc there, we were very interested in developing unbiased approaches to really look at the vast terrain of these interactions. And as many of you in the audience would appreciate, when we look at the interaction landscape of the proteins across the human proteome, which is about 20,000 proteins in the human proteome, you can appreciate that there is a huge zone here, which is called dense zone, what we want to call it. And these are interactions that have been repeatedly identified in the literature. One that comes to mind is p53-MDM2, for example, that people have been cataloging. So, of course, there's a huge need to be able to understand and uncover interactions in what we call this the sparse zone. And a lot of these genes that are in the sparse zones are genes that are implicated in Mendelian diseases and cancer. And so what our work was over the past decade to really establish a suite of assays, if you will, and I won't have time to go into the details, both in mammalian cells, yeast, as well as a number of in vitro assays that we've established in the lab to really discover and identify these interactions in this sparse zone, as I would show you here in the next slide, which is really now you can see when we carry out these systematic approaches, we can really populate this sparse zone, as I was showing you in the previous slide. And the hope is that over time, as we continue to populate these high throughput assays, we can really try to continue to identify these uncharacterized or previously unidentified or unappreciated interactions. And as a result of this effort, and this effort has been constantly evolving, in the previous map, we had uncovered a landscape of about 14,000 interactions among 4,000 proteins, a large number of which were not previously identified. And so, as I said, once we have this reference network of interactions, we then want to come back to the initial problem that I was alluding to, is how do we solve this, the black box that exists between the genotype and phenotype. And the first part in that process here is to really be able to find causal variants. As you know, like there are a number of genomes these days. Sequencing has become routine with major advances as well as the cost coming down. We have a large number of variants that are being identified across genetic diseases as well as cancer. And I was very fortunate when I joined MD Anderson to be part of the Cancer Genome Atlas and some of the other consortia efforts that have looked at whole genome sequences also across cancer patients. And so the idea is how we can leverage on our approaches to really identify which are the causal variants in the spirit of early detection, and then how do we characterize them. Of course, many labs across the world are doing this on a smaller scale, but how we can develop the technologies really to scale this problem and functionalize and contextualize these variants. And we've done a number of seminal projects in this, both in the context of rare genetic diseases, as you will see in the next couple of slides, as well as in the context of cancer. And so this project was a culmination of a large team effort that I was really engaged in when I was working with a great team during my postdoc years. And we called it the 1,000 Diseases Project, as I said. So we are kind of disease agnostic in this case, really taking at a broader look across genetic diseases, mostly Mendelian disorders. And as I said, we've established this framework where we can really assemble really thousands of these mutations that are being identified as we speak, and really assemble these libraries of these mutations, which, by the way, we made available to all researchers all over the community. And this has really helped the community. It's been a communal resource to really push the field forward in Mendelian rare diseases. And then the idea is that we can really use a suite of assays as part of our functional variomics approach, which begins with assembling this library, if you will, of large numbers of these mutations, and then characterizing them across a suite of assays, which I will describe very briefly, and happy to talk more over break and other opportunities during this meeting. And so as one of the assays that we used as part of this work, of course, we were looking at characterizing protein-protein interactions, as I had mentioned earlier. And we do this in a number of model systems, including yeast, as well as in mammalian cells. And using this approach, we can really go in and actually compare how do these mutations, in this particular case, a single point mutation really alters this network of interactions when you compare to the wild-type protein or the unmodified protein. And so using this approach, we very nicely showed as part of this work that using this approach, we can really distinguish these two different classes of mutations, if you will, one of which we call disease mutations, which are both mutations identified across a suite of Mendelian diseases, and then, of course, non-disease common variation that we identify from sequencing just regular normal individuals, quote unquote, as far as we know they are healthy. And there's a lot of effort that's been obviously going on in this regard, for example, with other genome sequencing projects, such as the All of Us and others that are coming down the pike. And using our approach, with the functional variomics, we were able to really classify these mutations into really two different bins: disease mutations, which were impacting which included a large number of alleles that impact protein-protein interactions. And on the contrary, if you look at these non-disease mutations, these were variants that were largely keeping the interactome network intact. In other words, they were not really altering any measurable protein-protein interactions. And this is a large fraction, which is about 92%, compared to when we look at the disease mutations. When you sum up the total of what we call these egetic perturbations and the quasi-nulls, these are a large fraction of these mutations that alter network protein-protein interaction networks. And in the interest of time, I will just skip through, but a large body of work that we did as part of this project, which was also going beyond just looking at protein-protein interactions, but rather take advantage of our high-throughput assays, but to look at interactions, for example, involving transcription factors, so in protein-DNA interactions. And I'll just skip this slide in the interest of time, but the idea was to be able to really take this functional variomics approach and integrate across these different assays. So looking at protein-protein interactions, but also looking at other assays that look at, for example, protein-DNA interaction perturbations. And in recent years, we've also gone in and looked at RNA binding proteins and how do these proteins alter protein-RNA interactions with a number of high-throughput assays that we've developed. So after I came to MD Anderson, I've been very interested in cancer biology, and as many of you in the room can appreciate, there has been a whole explosion of identification of these genetic mutations across a number of projects, as I've alluded to earlier, both at the level of many different cancers, which was the TCGA and ICGC, but also at the level of individual cancers, where people have gone in and cataloged the complexity of variants that exist across the entire plethora of cancer genomes. And so when I first started this work, of course, we started with a very defined subset of genes, as you can see on the histogram at the bottom. Many of these genes you will recognize. These are known kinases. Of course, a few of the tumor suppressors, very well studied over the many, many decades of work from many, many different laboratories across the world. But what struck our attention here was that even though these genes have been very widely studied, when we look at the landscape of mutations that have been identified in these genes, a large fraction of these mutations still remain largely uncharacterized. If you can see this pie chart on the top, sorry about the small lettering, but you can see the gray part really defines what the problem was at the time when we started this work, taking advantage of course, algorithms that, for example, OncoKB, that Memorial Sloan Kettering investigators at MSKCC have developed, as well as algorithms that we've developed at MD Anderson, as well as taking advantage of automatic text mining at PubMed. And looking for abstracts that have any evidence of any of these mutations being characterized at any functional level across the literature. And to our surprise, as you can see, close to about 80% of these variants still need assays and technologies to be able to really classify them as having an impact on cancer phenotype or not. And so as part of this work, which was, again, I was very fortunate to have collaborated with a whole team of investigators that I'll acknowledge at the end of it. We really assembled, again, taking advantage of our functional variomics approach to really establish this large library of these mutations and actually characterize the impact of these mutations in 2 different cell lines. And just at the end of this exercise, we can really classify whether a mutation is going to have a functional impact on a measurable cancer phenotype. In this particular case, we looked at whether it can impart better fitness or better survival to cells or poor fitness. And also, we looked at high-throughput assays to look at protein expression alterations, which we call the RPPA assay. So we can also measure the impact of these mutations on how they alter protein expression in an unbiased manner. And so just to make a long story short, just taking an example of this gene, EGFR, and in this particular case, a lot of seminal work, again, has been done in the literature, but what distinguishes us from other efforts is to really look at the suite of mutations across this entire protein, including, of course, the commonly studied hotspot mutations, but a large number of mutations that had not been previously characterized, and really assign function, if you will, to these mutations. And this is what we describe here as the first column in this heat map. And then the idea is that we can really take advantage of our unbiased approaches to really characterize these mutations that have been previously uncharacterized and provide these data, unbiased data, to these training algorithms to better classify these mutations and their impact of these mutations on function, so we can really facilitate AI in order to be able to better predict the impact of these mutations, which is still a big challenge in the field. And then, of course, in the particular case with EGFR, we also annotated and see where these mutations exist in the 3-dimensional structure of EGFR, and for example, in BRAF, and we found that mutations that were in fact having an effect on fitness tended to cluster in the 3-dimensional structure, as you can see in this particular case. And so basically, with the culmination of a large amount of work that we did over the course of time, of course, in cancer, we've also developed this algorithm which we call PHASMIC, in short, for functional assessment of somatic mutations in cancer, wherein we've actually collected information, of course, taking advantage always of information that exists in the literature. Of course, a number of people in the field are developing computational algorithms, but we are also providing our phenotyping data to these groups and interacting this back and forth that happens so that we can better predict with a great accuracy the impact of these mutations in the future. If you will, if you go in and type your favorite gene or its associated variant in Google, you should be able to see whether what has been assessed at the variant level for any given protein in the human proteome. And so just to give a glimmer, this is just, again, a small sliver of what we do in my lab, but we've also gotten very interested in, for example, looking at the impact of these mutations on phase separation and condensation, which is also a very emerging field in biophysics that is now being greatly appreciated in biology. And of course, as I will show you in the next slide, we're also taking advantage of some of the new technologies, for example, with CRISPR screens, to really identify rational combination therapies, in particularly with my deep focus on ovarian cancer, as Dr. Sood had mentioned. And so I've been really fortunate, to be working at MD Anderson, where we have this incubator where we can really interact very closely with clinician scientists like Dr. Sood, and I've been very privileged to have been working with him over the past several years. I never had a very deep understanding of ovarian cancer. It struck me personally when my mentor passed away tragically very quickly after she was diagnosed with ovarian cancer. And it was really in the spirit of all these people. We have a network of these friends from Sue's lab that we actually teamed up, and they said, you're at MD Anderson, we need to make a dent in this disease in some form or shape. And I took it to heart, and I started this work. And so this award is a really great culmination for some of the hard work that we've done in my lab, over the last 6 years to really work on ovarian cancer, hoping, with some of the approaches, as Dr. Sood alluded to in the introduction, to really make a dent in this disease. And in that effort, some of the work is just coming out later in the year, last year. And then we have, of course, ongoing work that's going on collaboratively with Dr. Sood. And so the idea there is to really be carrying out these drug screens to identify novel combination therapies with the standard of care, which is PARP inhibitors right now in high-grade serous ovarian cancer. And we're using a number of both experimental approaches. For example, one of the work that was just published recently took advantage of this assay RPPA, which is really looking at protein expression alterations on a handful of proteins, few hundred proteins that my previous chair, Dr. Gordon Mills, has developed in his lab. And then, of course, we are also constantly evolving, in the spirit of this meeting, developing computational algorithms to identify novel drivers of HR deficiency in ovarian cancer, and really to see how we can combine some of these novel targets with PARP inhibitors to really help patients with high-grade serous ovarian cancer. And we have been very fortunate to have been just granted an award of funding with Dr. Sood to really continue this work to develop AI approaches to prioritize these drug combinations in high-grade serous ovarian cancer with the Ovarian Cancer Research Alliance and Microsoft just this past year. So with that, I would really want to acknowledge, as I said, a lot of the work that we do is really team science and team effort. I'm just listing part of my lab that has been instrumental in helping with some of this work. We, of course, do a lot of this work collaboratively across many different institutions, and some of the work that I didn't show today, but I've been very fortunate to have collaborators. Part of this work was very closely done with my partner and my husband, Stephen Hui, who runs a computational biology lab at UT Austin. And of course, I'm incredibly grateful to the leadership at MD Anderson, in particularly the chief scientific officer, Dr. Julio Dareta, and Dr. Eyal Gottlieb, who's here at this meeting in attendance, who have been really helping us push the avenues for discovery science. And then, of course, I will forever be grateful to Dr. Gordon Mills, who had the vision to get me to MD Anderson back in 2016. And then I think you will hear this also in Ben Dineen's talk tomorrow. We had a common friend, Ken Scott, who used to be at Baylor College of Medicine, who unfortunately succumbed to cancer. So I have a really, I'm really grateful to Ken and learned a lot from him when I first started my lab at MD Anderson. And of course, a number of computational folks, Ken Chen, who works at MD Anderson, and a number of people, who are at Baylor. We interact very closely, and I'm very fortunate to have been funded through a number of initiatives here listed. CPRIT being the top of the list, I really think it really has had a transformative impact on my research, and I'm a strong advocate for team science, and really privileged to be part of a number of these consortia, one of which just started a couple of years ago with the NHGRI, which is the Impact of Genetic Variation on Function Consortia. We are partnering with these two investigators at the University of Pittsburgh to try to understand the impact of non-coding variation, going beyond coding variation now in my future work. And last but not least, I also would like to thank my family. I've been I was very lucky that I actually met my husband when we were graduate students, and he was a senior in the lab at University of Iowa. And we have a 8-year-old Kevin. We have been very fortunate and blessed. And also my parents here with actually our collaborators, where we were having dinner when we were back home, this past winter. So really privileged again here to present my work, and thank you for your attention. And one last thing, which I think would be amazing if we are always recruiting people in our lab, and I would very much appreciate if you know of people looking for positions to refer, if there are opportunities. With that, I'll be happy to take questions. Thank you.
Okay, we have time for a couple questions, so just go on up to the mics and ask.
It's a fantastic effort, and the amount of knowledge is coming from it. And you hinted during your talk, but I would like to elaborate a little bit more. How far can we go with just protein-protein interaction? I know you're doing also DNA. How much that information is sufficient? And also, you're talking about all possible single mutation. How about epistatic effects that can come on disease? How do you handle? Can you do that sort of come from your data? Can you elaborate on that a little bit?
Yeah. No, really great question. I completely agree that, that's why I want to allude, and because of the length of the talk, I couldn't really go into the details of some of the other assays. I definitely think protein-protein interactions in a set of assays is just one part of the problem. The idea is to really globalize this across many different phenotypes and assays that we are looking at. In my current funding that I have with the NIGMS, for example, a grant that we have, we are also very interested in looking at combinations of mutations to measure allosteric effects, and that's another project that we've been developing in the lab, really to understand combinations, as you said, because many mutations don't happen in isolation. And more importantly, where we've gone and taken the field forward has also been in the space of cancer translocations, for example, these larger structural rearrangements that happen in the cancer genome, and we've been funded actually as part of CPRIT to really look at these larger structural rearrangements and how do they impact cancer phenotypes. So more to come, I guess, and we are definitely thinking.
I agree, and that's wonderful, and I think you're really pushing that direction. But looks like even just still the level of single mutation or just on protein penetration, a lot of information comes out. So I was curious in a sense to know base. Yes, we can always increase the problem, but it's basic. Now that you have this database, is that a good screening, just stay on PPI and?
Yeah, so it has a really amazing predictive power, right? I mean, with the data we have in hand, but we're always still want to make it better. And we're making this data available to, of course, computational biologists who are developing these algorithms. And we develop these together as part of this back and forth that happens to be able to train these algorithms, as you know, because a lot of the data that's going in the training of these algorithms is highly biased. And so we really want to constantly evolve the training that happens with these algorithms so that we can really predict accurately what's the impact of the mutation. So
thank you. Congratulations.
Thank you.
So we can take one more short one.
Thank you for the interesting talk. You had one of your slides about tobacco, and that triggered the thought in me. Have you explored the role of the exposome in many of these cancers? And I would be delighted to collaborate with you.
Yeah, it's a really great question. As I said, like today, I just summarized the work that we've done, if you will, under one condition with a certain set of assays. Now we're adding more complexities and adding more stressors, and to see how these interactome networks rewire dynamically with a number of stressors. And so there's more work in that direction going on with, like, for example, DNA damaging agents in the lab as well, in the context of lung cancer, which is, again, supported by one of the tobacco initiatives that we have in our department. So happy to talk more and look forward to interactions.
Let's congratulate and thank Dr. Sahni one more time.
Thank you. Appreciate your help.
Oh, yeah.